Hello, everyone. Welcome to our monthly Science of Alt Protein seminar. My name is Amy Huang, and I'm the Associate Director of Scientific Ecosystems at the Good Food Institute. I realize that some of you might be here for the very first time. So, to those of you who are here for the first time and interfacing with GFI or with this scientific community, welcome, welcome. The Good Food Institute is a nonprofit think tank that is 100% driven by philanthropy, and our goal is to really work together to build a world where alternative proteins are no longer alternative. So, if you're looking to take a deeper dive into science of plant-based fermentation or cultivated meat technologies, if you're interested in seeing how GFI might be able to support your efforts, please check out our open access resources at gfi.org/science, and don't hesitate to reach out. Again, I'm joined by Dr. Victor Ujor. Dr. Ujor obtained his bachelor's degree in applied microbiology and brewing from the Enugu State University of Science and Technology in Nigeria. He received his master's and doctoral degrees from the University of Westminster in London in applied molecular microbiology and biotechnology. And after a postdoctoral experience at Ohio State University, he served as an assistant professor at the same institution before joining the University of Wisconsin-Madison, where he is now as an assistant professor in the Department of Food Science. And today we're going to be talking so much about fermentation and the central role that it plays in this quest to sustainably and economically produce and formulate alternative proteins in a way that actually allows us to create direct replacements for conventional animal-derived proteins. I won't say too much more because Victor will really take us through today. So we'll dive into the exciting results of Victor's research. I'm really excited to learn alongside all of you today. And with that, I will pass it over to Victor. Thank you so much for being here with us today. And we look forward to learning about all of your research.
Thank you very much, Amy, and thanks a lot to Good Food Institute for having me today, and thanks to everybody that's watching online. It's a pleasure to talk about sustainable carbon and nitrogen sources for fermentative production of alternative proteins. When we mention fermentation, often what comes to mind is beer and wine and cheese and all that. And all of that is good and dandy fermentatively speaking. However, today we're looking at using fermentation to produce protein-rich biomass, but more specifically and importantly, we're looking at alternative proteins via precision fermentation, not just to synthesize specific alternative proteins, but also to synthesize the ingredients used to formulate alternative proteins like vitamins, fats, and pigments. Often when we say this, something comes to mind, the hemoglobin or heme that was synthesized and used by Impossible Foods. However, one thing we need to bear in mind in terms of producing alternative proteins is that if we don't think about the sustainability of the feedstock we use to make them, the sugars and nitrogen sources, then we're likely to end up in the same trap that animal protein is. Of course, is well documented, the impact of animal protein production on greenhouse gas emission and climate change. So there's growing interest in precision fermentation, there's growing interest in alt proteins. Also, there's a growing human population as well. If you put all this together, we're going to have a lot more mass of feed in the coming years. And that means we have to produce more proteins fermentatively using precision fermentation. And the consequence is that if we're not doing it sustainably, that could somewhere down the road harm the environment in one way or another. Right now, to produce alternative proteins fermentatively, we use refined sugars or corn sugar or cane sugar. These all work, but the problem with them, or the problems with them, are both economic and environmental. From an economic standpoint, they're not the cheapest form of sugars for fermentation if you want to produce something massively, especially something we're going to eat. From an environmental and logistical standpoint, you have to think about the fact that corn sugar and cane sugar as well are grown specifically for food supply, and if we use them for fermentation, then we're disrupting that supply. Probably we'll have to do more, fell more trees, create more land to be able to arably grow the crops and get the sugars. So we have to think about something more sustainable, something that's cheaper, always available, and doesn't disrupt food supply and doesn't leave a lot of footprints environmentally speaking as well. While we know a lot about carbon sources for fermentation, not many of us think about ammonium as a source of fermentation, nitrogen sources. We cannot ferment without nitrogen. This is because just like you have to eat a balanced diet, microorganisms need a balanced diet as well. Because if they don't have nitrogen, they can't produce proteins, they can't make nucleic acids, they die. So we need to supply carbon in a particular ratio with nitrogen sources. And some organic nitrogen sources work very well, but for some organisms, they do require inorganic nitrogen sources, typically in the form of ammonium salts. And today, there's only one source of ammonium in society, the Haber-Bosch process. It's not sustainable by any stretch of imagination. I'll touch on that a little bit as we go forward. Collectively today, I'm going to talk about the challenges associated with traditional carbon and nitrogen sources, some of which have already been highlighted. I'll look at potential solutions in regards to some of the work we're doing in my lab and the case studies as well that will stem from that. I talked about ammonium salts. Ammonium salts are very important sources of nitrogen in fermentation. To produce ammonium via the Haber-Bosch process, the singular source of ammonium in human society, you need to combine nitrogen and hydrogen. You can see there hydrogen comes from natural gas. Of course, that is not sustainable. It has a lot of environmental footprint and contributes to climate change. The other side of it is that you need to ramp up the temperature to around 450 degrees Celsius and 200 atmosphere for this process to work. The consequences are it consumes a huge amount of energy. As a matter of fact, the Haber-Bosch process accounts for 1.8% of global energy supply, consumption rather, just one process. 3% of global fossil fuel consumption, and it generates over 560 million tons of CO2. That's 3% of global output, just one process. So it's easy for us to use ammonium salt in fermentation. If we think about it very critically, where it's coming from, are we really making that protein sustainably if the source of the ammonium salt used in the fermentation process is not sustainable in itself? So for that reason, my lab started looking at nitrogen fixation. For those who don't know nitrogen fixation, I bet most of us do, but it's simply a process where microorganisms that coexist with plants do fix nitrogen from the atmosphere into ammonium, which they use and share with the plant. The atmosphere contains about 7 to 8% nitrogen, so it's abundant, it's renewable, it's always there. These organisms have evolved the capacity to fix it and use it. And there's been a lot of interest in nitrogen fixation, but specifically from the standpoint of plant production, not from a fermentative viewpoint. So we started asking the question, can we fix nitrogen in fermentation the way we actually think about it from a plant production perspective? Can we grow organisms that can ferment food or make proteins for us or any other product of interest, and at the same time fixing nitrogen? That would be a lot more sustainable. I'm going to go a little bit more into this over the course of this presentation. For now, let's look at carbon sources first for precision fermentation. Beyond your corn sugar, cane sugar, and refined sugar, the other major source of sugar in society is lignocellulosic biomass, the most abundant renewable material on the planet Earth. These are things like corn stover, forest residues, wheat straw, and what have you. The beauty of these is that you don't grow them specifically for fermentation. There are side streams, waste products from the production of other crops. So you're not directly impacting the environment just to make your protein. So these wastes themselves eventually get burnt if we don't use them. Can we find applications for them from a precision fermentation standpoint? They are cheaper, they are renewable, they are more abundant. However, before we look at further how we can extract sugars from them, we have to highlight that lignocellulosics do have the challenge of likely containing aflatoxin. These are fungal-derived carcinogens you're likely to find in plant materials. However, the process with which you derive sugars from lignocellulosics has a very high tendency to remove or eliminate aflatoxin. For instance, this is what your lignocellulosic molecule looks like. You have lignin here, which protects the sugar-bearing polymers cellulose and hemicellulose on the inside. For us to get access to sugars in the cellulose and hemicellulose, we have to crack open the lignin. So we raise the temperature, not quite as much as we do with Haber-Bosch process. Here, about 115 degrees Celsius is enough to break this in the presence of alkaline or acidic solution. So at high pH or low pH with high temperature, 115 degrees Celsius, you crack open the lignin matrix, then you can get access to the sugar-bearing polymers. With fungal or bacterial-derived enzymes, you hydrolyze them and you get your sugars. If you've worked with this before, after you've done pretreatment, which is ramping the temperature at high or low pH, And then you treat with enzyme, you obtain your sugars. If you smell it, it smells like pure raw sugar, has that syrupy smell to it, full of sugars. The moist heat of this process, the high or low pH combined, typically destroy aflatoxins. So the risk of having aflatoxin carried away after this process is extremely low. However, in addition to that, after precision fermentation, you make a protein or any of the product you're going to use to formulate the protein fermentatively, you will purify before you use it. These purification processes are often very specific, which allows you to selectively remove anything else and collect your protein or other product. In that case, you have a high chance of eliminating the carryover of aflatoxin. The real problem, though, with lignocellulosics is that when we break cellulose and hemicellulose down to get sugars, the conditions that we use to do that also enhance what we call dehydration and degradation reactions of the sugars, which leads to the production of aldehydes. Examples of furfural, hydroxymethylfurfural, syringaldehyde, phenolic acid, and what have you. But these two I'll focus on the most because they're usually the most abundant, furfural and hydroxymethylfurfural. They're not toxic to us. In fact, some food we eat contain furfural and HMF as well, but they are toxic to microorganisms. They can't stand them. They cause DNA damage in microorganisms. They cause membrane damage as well. They inhibit enzymes, particularly glycolytic enzymes. These are the enzymes that need to actually feed on sugar. So if they can't feed on sugar, they die. But most importantly, they cause what we call redox imbalance. Microorganisms cannot function enzymatically speaking without redox balance, which is NADPH that they use to convert certain products to another. If you don't have a balance between NADPH and NADP or NADH and NAD, the cells will die. So collectively, these results in the arrest of fermentation. It starts and the cells will start fermenting, then they die because of the toxicity of furfural, HMF, and the other inhibitors I talked about. So the real problem with lignocellulosics is really how can we overcome these inhibitors? Because you will have your sugar, you have a small concentration of the inhibitors that prevent you from using a high concentration of sugars. If we can engineer or identify microorganisms that allow us to circumvent or degrade or utilize these inhibitors, then we can efficiently ferment the sugars that come from lignocellulosic materials. So most of my work is in the area of overcoming this challenge. One thing we can do is to isolate or engineer a bug that has the capacity to use these inhibitors as a carbon source. They can catabolize them. The other thing are identifying organisms or engineering ones that can detoxify them. We'll look at detoxification a little bit more. But in terms of catabolism, which is using the inhibitors as a carbon source, that will completely eliminate them from that environment. We could have a two-step approach, whereby you engineer a bug or identify or screen and find a bug that you can inoculate into the lignocellulosic biomass hydrolysis, the sugar solution you obtain after you break down biomass material to obtain sugars. You can inoculate the organism, it grows on the inhibitors selectively and removes them using little to no sugar, and then you can pass your sugar to the next stage, whereby you inoculate your organism for precision fermentation to make a protein or any other target protein or product of interest. The other thing you could do is to use one bug to achieve that, whereby one organism can actually do detoxification by catabolism and at the same time make your product for you. Now, let's look at detoxification before we go any further. What happens with detoxification rather than catabolism is, for instance, if you have 5.5 grams per liter furfural or hydroxymethylfurfural HMF, organisms will reduce them chemically speaking to their respective alcohols, furfural alcohol and HMF alcohol. That process of reduction from aldehyde to alcohol consumes a lot of NADPH. This is why these compounds are quite challenging to microorganisms. Because they are toxic, they will abandon every other metabolic process and focus on detoxifying these compounds. In doing so, they consume a lot of NADPH. It throws their redox balance out of whack and the cells will die. So detoxification doesn't often work. At a low concentration, that is fine. At a high concentration of inhibitors, which you typically find in biomass hydrolysates, it doesn't work. So most of my work as a result is focusing on catabolism, engineering and identifying microorganisms that we can tweak further to remove all these inhibitors by using them as carbon source alongside the sugars. How do they do that? The organisms that do it naturally, what they do first is to convert all that HMF to HMF alcohol. HMF alcohol is significantly less toxic than HMF. So they can slowly pass it back to HMF and through this pathway to ferric acid and feed it into the TCA cycle, thereby using that as a carbon source. The important thing here is that at any point in time, you have a low concentration of toxic HMF because they're banking it as HMF alcohol, then using it slowly. The same thing happens with furfural. They convert it very rapidly to furfural alcohol, which is significantly less toxic, and then they slowly pass it back to furfural at low concentration and feed that to ferric acid. Ferric acid is very important. When you grow a bug in biomass hydrolysis and you take ferric acid, it does suggest that there's a very strong likelihood that that organism uses that inhibitor as a carbon source. So for this purpose, we're working on two particular organisms, Cupravitis basilensis and Penibacillus polymyxer. Both of them we found can use these inhibitors as carbon source. I'll present some of our results and some of the things we're trying to do going forward with them. First of all, let's look at C. basilensis. It does use inhibitors as carbon source. We found a strain, and we've been studying that strain for quite some time now. Our ultimate goal is when you do biomass hydrolysis and pretreatment, you have a mixture of sugars and inhibitors. And there, can we selectively use C. basilensis to remove the inhibitors, and you'll be left with a sugar-rich solution to which you can feed any organism in fermentation to make your target protein or any other product. So to do that, first of all, we had to look at how the strain we're working with grows on different concentrations of furfural and HMF from 0.3 to 2 grams per liter. If we look over here, this is furfural, this one is HMF. You can see that it does grow on all of these concentrations except the last one, which is 2 grams per liter over there. Before I go further, I'd like to mention that it does grow on 2 grams per liter if there's glucose supplied. As a sole carbon source, it struggles with that. It's a less palatable carbon source than sugar. If you think about it, there are things I like and things I don't like. When I'm fed things I don't like, particularly, I eat them sparingly. It's the same thing with this organism. On the other hand, with HMF, it even grows better at 2 grams per liter HMF. It does like it a great deal. So we saw that it can grow on both of them to different degrees. Then we measured the disappearance of these inhibitors. First of all, from 0.5 to 2 grams per liter, you can see over here, clearly at 0.5 and 1 gram per liter, C. basilensis grows on furfural. The furfural concentration drops significantly in a few hours. And you can see the concentration, the blue one, which is the furfural alcohol, goes up and comes down. That means it's consuming it, especially if you see ferric acid, and clearly you can see ferric acid appear and disappear there. If you look at HMF, for instance, at 2 grams per liter, clearly the concentration drops significantly, and the same thing with HMF alcohol and ferric acid coming up and disappearing, which confirms that it does use both of them as carbon source, but more efficiently HMF than furfural. So we looked at other inhibitors. Beyond these, you have other inhibitors you actually obtain when you break down biomass hydrolysis, biomass materials, things like para-cumaric acid, vanillin, vanillic acid, 4-hydroxybenzaldehyde. So we grew the C. basilensis in the presence of different concentrations of these compounds. You can see it grows in all of them, the least one being 4-hydroxybenzaldehyde. And if you look at the concentrations of these inhibitors, they're all reduced in about 48 hours. Then we decided to combine them together. Does it grow in mixtures of furfural and HMF? Clearly, it does grow on both of them. That's the growth curve there. If you go over here, look at furfural and HMF decreasing significantly within about 24 hours. The most interesting thing, you can see the ferric acid concentration rising and dropping, which is a confirmation of its ability to use both of them collectively as carbon sources. Then we combined vanillin, vanillic acid, and para-cumaric acid. Again, the same thing. It does consume all of them as concentration drops significantly within about 3 days. Then the next thing is, if it does use these inhibitors, the chemically synthesized ones, what about the ones you derive after you break down your biomass hydrolysis? We work with a grassy biomass material called Miscanthus giganteus. So we obtained it, dried it, ground it down, and put it through the pretreatment process, hydrolyze it, obtain this syrup full of sugars, as I mentioned a while ago, also contains inhibitors. And we looked at how C. basilensis would grow in its presence. First of all, the first thing we wanted to see is what happens to the inhibitory compounds over time. That's furfural all the way down to 4-hydroxybenzaldehyde. They were all completely removed, 100% removed from that biomass hydrolysate, except cinnamaldehyde and ferrolic acid. So it was exciting that it does remove all of them. Then we asked the question, if it removes all of them, almost all of them, I should say, from the biomass hydrolysate, can we now go further to ferment this into any product of interest? We tried that. We used Clostridium beerinckii as a proof of concept. This organism produces butanol, acetone, and ethanol when it grows on any sugar. So we looked at 60% biomass hydrolysate, in this case, Miscanthus giganteus hydrolysate, 80% and 100%. How does Clostridium beerinckii grow on this hydrolysate after detoxification using C. basilensis? 60%, you can see in all cases, from 80% to 100%, Clostridium beerinckii was able to grow significantly better than in the non-detoxified biomass hydrolysate. Then the ultimate question is, does it make a product? If it makes a product, it suggests we can actually use this to synthesize proteins fermentatively. 60%, 80%, 100% acetone, butanol, ethanol combined, you can see much better than the undetoxified one. Something we noticed here is that if you look at the concentration of acetone, butanol, ethanol produced at 100% biomass hydrolysate relative to 60% and 80% is much less. So clearly we think, if you go back a bit, that the residual inhibitory compounds, in this case cinnamaldehyde and ferrolic acid, are really exerting a significant effect on both growth and the production of acetone, butanol, and ethanol. So this is something we're doing further. What can we do to enhance the ability of this particular organism after pretreatment and hydrolysis to be able to remove all the inhibitors? That's one, with C. basilensis. If that works, will we be able to make more products? Can we deploy this in precision fermentation as well going further? Before we go any further, I'd like to highlight the summary of that graph the graphs over there. If you look at the growth pattern in the detoxified biomass hydrolysate and undetoxified one, clearly the detoxified one, we got much better growth. Look at acetone production more, ethanol production more, butanol production more, and that applies across the board for all the parameters we measured. In fact, ethanol was the only product that the Clostridium organism produced after we grew it in the undetoxified biomass hydrolysate. The problem here, though, was that while C. basilensis removed all the sugar, all the inhibitors, almost all the inhibitors in the biomass hydrolysate, it also used about 50% of the sugars. So that's a bummer in the sense that you want to selectively remove the inhibitors and have most of the sugars left behind for precision fermentation along the line. In this case, the organism was too greedy to remove most of the inhibitors, quite a chunk of the sugars we were interested in. We also think that that might have played a part in the growth patterns of the organism as well as the products produced, because we were expecting more than what we obtained in terms of the acetone, butanol, and ethanol produced. And that brings the next question we're looking at currently. I have someone working on, can we now engineer this bug in such a manner that it will selectively and robustly use inhibitors and use very little, if at all, any of the sugars? So we can knock out the sugar utilization pathway, or at least limit the capacity of this organism to absorb and utilize glucose and the rest of the sugars, while enhancing its capacity to use the inhibitors. And that's something we're looking at as a means to clean up biomass hydrolysate for future fermentation experiments. The other thing we've looked at is the Penibacillus polymyx, so the second organism I talked about. In this case, we're thinking of obtaining our biomass hydrolysate to get the sugars. We don't have to pre-clean up with a particular organism. We want to use one bug for the whole thing, clean up the biomass hydrolysate, and make our product of interest. And why are we working with this organism? There's a lot of advantages to working with Penibacillus polymyxa. One is generally regarded as safe, and I have to say C. basilensis is very safe, non-pathogenic as well. But a lot's been done with Penibacillus polymyxa over the years. It's very safe, it's non-pathogenic, it's a soil-inhabiting organism, it's nitrogen fixing as well. I'll get to this as we go further, which raises that potential. Can we remove inhibitors from the biomass hydrolysate, fix nitrogen, and make a protein down the line? That would be superior economically speaking and environmentally speaking as well. Also, this organism produces acetone, it produces 2,3-butanediol. These are both used in food formulation, so they're very good and can be extracted as co-products if you're making a protein. But most importantly, this organism has a robust protein secretory machinery. So you can take a protein of interest, put it in the genome, it makes that protein for you. Just like most bacillus species, it behaves like that. So that's our major interest. Can we establish this organism for making proteins? First of all, we have to demonstrate that it can fix nitrogen in a fermentative environment. Secondly, it can efficiently remove the inhibitors in the biomass hydrolysis. The other issue, or the one major issue with this organism, is that it produces exopolysaccharides. They're not dangerous, they don't hurt anyone, they don't have any pathogenic issues we know of, but they're yucky, so to speak. But most of all, if you're making a protein with this organism and you have the protein line in the system with things, you can see all the materials lying around here and there. These are all exopolysaccharides. It's called levain, the major one produced by this organism. It will add to your cost of production, whereby you have levain in the system as a polysaccharide and your protein. You have to separate them. So that increases the process and cost of recovering your product. So we thought, if we're going to use this organism to make protein down the road, we don't want a situation whereby exopolysaccharide is increasing the cost of protein production due to recovery costs. So we went further to think about how to remove the capacity of this organism to produce levain, the polysaccharide I talked about. So we fully sequenced the organism after sequencing the whole genome. We mapped it, went in there, and found a gene called levain secretase gene. We decided to knock it out using homologous recombination, whereby we replace it with an erythromycin resistance gene, pocket into the genome. That will, during recombination, we insert it into that particular gene, truncates the gene, and in place of that, it expresses erythromycin resistance, and we can select that on an erythromycin plate. We did that, selected, sequenced again, and confirmed that the strain we're looking for was there. Does it produce levain? If you look over here, this is your wild type, your native natural organism. The blob you see up there and down there, this is your levain, the exopolysaccharide. It produces lots of it. And the one that we engineered, where we knocked out the levain secretase gene, you can clearly see that the levain, only a very small amount is produced. It's almost gone. So we now have more like a canvas where we don't produce this exopolysaccharide. We can now use this to make proteins, and we don't have that added cost of separating the protein and the levain, which is a very yucky material to work with from a fermentation standpoint. So the next thing is looking at biomass hydrolysis. How will this organism grow in biomass hydrolysis? Specifically, we work with wheat straw hydrolysate here. We took the wheat straw just like with the miscanthus. We put it through 115 degrees Celsius in the presence of acid, cracked open the lignin, used enzymes to obtain the sugars, and this is what it looks like: 4.5 grams per liter arabinose, almost 56 grams per liter xylose, and about 39 grams per liter glucose. So that was exciting. We got all the sugars. But if you're using biomass hydrolysate, apart from inhibitors, the other thing you have to contend with is that you have a mixture of sugars. If you have a mixture of sugars, what microorganisms often do is to selectively feed on them, because it's energetically expensive to try to eat three things at the same time. So they make the enzyme they need to feed on usually glucose. This is called carbon catabolite repression. The presence of glucose will inhibit all the enzymes required to grow on xylose and arabinose in most organisms. While that should be fine and dandy if they would go on to feed on xylose and arabinose after they've used the glucose. It doesn't always pan out like that. For whatever reason they get, I would say, metabolically tired, or some of the products they make kill them, that they feed on the glucose and they don't use the other sugars you obtain at the end of the process. And that is a travesty, because you spend all that money, obtain the sugars, use only a small fraction of it. So our first goal here was to establish whether this organism can use all the three sugars. If it can't, can we engineer it to use all three sugars simultaneously as much as possible? So thankfully, when we grew it in the presence of glucose alone, xylose plus arabinose alone, and all three of them combined, glucose, arabinose, and xylose, in the same ratio as you would find in wheat straw hydrolysate. So let's focus on these two panels. If you look at the growth in the presence of glucose and growth in the presence of glucose plus arabinose plus xylose, clearly very similar growth patterns. It doesn't seem to have a major drop-off. So that was exciting for us. And one way to determine whether this is ideal for this organism is its natural product, which is 2,3-butanediol, does it produce enough of it in the presence of all three sugars combined? And that was what we saw. It produced about the same amount as it did in the presence of glucose alone. So does it use all three sugars together simultaneously? That's the important thing we wanted to establish here, that is not subject to carbon catabolite repression. So we looked at that. Xylose, you can see the concentration of xylose dropping down rapidly. The same thing with glucose. Of course, arabinose was simultaneously used right there. Now, this is one of the very few organisms we work with that are not naturally subject to catabolite repression. In fact, when we sequenced the entire genome and mapped it, we did not see the key elements of carbon catabolite repression. So this organism is capable of using all these three sugars simultaneously. Again, that's an exciting thing that we found out from this study. So if you look at the summary of the previous graph I put up there, the growth in the presence of glucose, 8.2 OD, 8.1 OD with all three sugars combined, about the same. You look at the product, in terms of 2,3-butanediol produced 32 and 32 grams per liter. So it behaves the same way in the presence of all three sugars combined relative to just glucose alone. That's very exciting for us. And the big question afterwards is, can this organism detoxify the inhibitors present in wheat straw hydrolysate? We looked at all the inhibitors we could search for in the hydrolysate. We found most of them, especially the big one, furfural, which is usually the most abundant, about 3 grams per liter present in the wheat straw hydrolysate. So the first thing we had to do was to grow it in the presence of the wheat straw hydrolysate, 60, 80, and 100% as well, relative to glucose control. Again, I'm going to focus on just these two panels here. You can look at the growth pattern. It grew better in the presence of all the wheat straw hydrolysate concentrations we studied relative to glucose. What about 2,3-butanediol production? Again, it did well with 60 and 80, similar to glucose, except 100% wheat straw hydrolysate. So we think there's a bit of residual inhibitors that are directly affecting the production of 2,3-butanediol. But exciting that at least at 80%, this organism is capable of producing the same amount of product as in the control glucose medium. So one of the things we're looking at is how can we engineer this bug a little bit more so that we can produce the same amount of the product, especially if you're trying to make a protein in this bug going down the road. If we open up that graph a little bit and look at the growth patterns, again, grew better in the presence of wheat straw hydrolysate, irrespective of the concentration studied relative to the glucose. You look at the maximum 2,3-butanediol produced, similar between the glucose and the 60% and 80% wheat straw hydrolysate. Now, we have to look at the inhibitors. What happened to them in the wheat straw hydrolysate? Clearly, we're seeing right here, if you look at HMF, hydroxymethyl furfural, at 60%, 80%, 100%, completely gone, 0, 0, and 0. Exciting. It removes all the HMF present in the medium. The other thing that was completely removed during the fermentation was syringaldehyde, gone all through across the board. Fumaric acid as well, gone all through across the board. However, that was not the case with furfural. At 60 and 80%, all the furfural was gone. At 100%, we saw residual 278 milligrams per liter furfural left, which we think accounts for that lower production of the target product in this case. And of course, other inhibitors like syringic acid and vanillin were still left behind. So this presents an opportunity for us to do further research on how to improve the capacity of this organism to be able to remove all the inhibitors as much as possible for future fermentation experiments. But does it use these inhibitors like HMF and furfural as carbon source, or does it detoxify them? That was the next thing we had to ask ourselves. So we grew penibacillus polymixer in the presence of furfural and HMF as sole carbon sources. First of all, 0.5, 1, and 2 grams per liter furfural. And we searched for fluoric acid, which we didn't find, suggests it doesn't use furfural as a carbon source, but we did find furfuryl alcohol. Also, we compared it to growth and glucose. If you just follow this graph there, the triangle, that's just glucose. And you look at the optical density in the presence of furfural, which is this one, not particularly high, the same over there, the same over there. And again, like I said, we didn't find fluoric acid, but furfuryl alcohol, the blue one, did come up, didn't significantly reduce afterwards, telling us that it's simply converting furfural to furfuryl alcohol, which is less toxic and doesn't use it as a carbon source. How about HMF? If you look at the growth on 0.5, 1 and 2 grams per liter HMF, that's the growth on glucose. That's the growth on HMF 0.5 over there, 1 and 2 grams per liter. Clearly, it does grow on HMF as common source in terms of OD alone, similar to the same optical density we obtain with glucose, albeit at a much longer period of time. Again, HMF is a less palatable sugar or carbon source than glucose, so we expect to see that sort of delay if it grows on any of these as its sole carbon source. But the exciting thing is that across the board in all these graphs, you can see ferric acid was detected. Not just detected, it comes up and drops in concentration over time, which means the cell is producing and then utilizing it, confirming that this organism can utilize HMF as a carbon source. And if you look at furfuryl alcohol, if you over there, that's HMF alcohol, which is the triangle there, comes up, goes down, same as your ferric acid. So after sequencing the genome of the organism, we map it as well, and we saw the enzymes responsible for its ability to use HMF as a carbon source. And that presents us with a lot of opportunity for further research. But before I go to that, if you look at this graph simplified, at 2 grams per liter HMF, similar optical density, much longer period of time to grow on HMF. And you look at the pattern of furfuryl reduction, clearly over time, used up all of it. And you look at furfuryl alcohol comes up, drops completely, and ferric acid is produced and then reabsorbed over time. Then we go over to furfuryl. We don't see any ferric acid, further confirming it doesn't use it as a carbon source. However, it does convert it only to furfuryl alcohol, which stays constant over the course of the fermentation process, and furfuryl drops for a period of time and then stops. So it doesn't use this as a carbon source. And based on that, we're excited that it does use HMF as a carbon source. We've identified the genes responsible for using HMF as a carbon source. We've also identified the genes for detoxification of furfuryl to furfuryl alcohol. And what we're doing now is to use protein engineering to structurally enhance the capability of the enzymes for HMF catabolism to be able to catabolize furfuryl catabolism. So you look at furfuryl and HMF, they're very similar in structure. The only difference here is a hydroxyl group over there. So if they're that similar, and it has the enzyme repertoire to be able to use HMF as a common source, we can tweak the enzyme, the binding pocket of the enzyme, to bind more tightly and hopefully catalyze the utilization of furfuryl. And that's an ongoing research in our lab as well. Hopefully we can obtain a strain that doesn't just use HMF as a carbon source, but also can use furfuryl as a carbon source. And hopefully we can remove most if not all the inhibitors in the biomass hydrolysate towards precision fermentation using lignocellulosic biomass hydrolysate with this organism. Finally, I'd like to touch on nitrogen. Most of us do a lot of fermentation with ammonium salts. We throw it in there, the cells grow well. They need that to make proteins and nucleic acids. But the source of nitrogen I mentioned is not sustainable, it's not environmentally friendly, it's the only one we've got at the moment. But if we're going to make proteins via precision fermentation, we need to think about the overarching sustainability of that process going down the road. However, the environment contains a lot of nitrogen that we can fix if we have the right organism. So if you grow an organism in an anaerobic environment, often you obtain this anaerobic environment by flushing out oxygen with nitrogen. So that nitrogen, which is cheap, can we fix it using the microbial bugs present to be able to make nitrogen and grow? So again, we're working with Penibacillus polymyxa. We do know that most Penibacillus species can fix nitrogen. The strain we're working with, we had to confirm that. So what we did was to starve it of ammonia, grow it in an environment full of ammonia, ammonia rich, and another one, ammonia deficient. How does it behave in ammonia deficient environment relative to the ammonia rich environment? So if you look over here, we grew the cells in the presence of 8 grams per liter ammonia, another one 1.5 grams per liter ammonia. At the end of fermentation, we measure the concentration of ammonia, and you can see clearly it reduced significantly in the ammonia rich environment. And over there was no reduction in the concentration of ammonia. This could mean that the cells died, they didn't grow, the ammonia stayed constant per se, or they did fix nitrogen. The only way to find out is to measure the growth curve, and clearly we look at the growth curve there. In ammonia deficient environment, we saw it got slightly better growth than ammonia rich environment. And ultimately, if this is the case, it should be able to make its natural product, which is 2,3-butanediol, and clearly it produces about the same amount of 2,3-butanediol in an ammonia-deficient environment relative to the ammonia-rich environment. So indeed, as far as we know, this is the first demonstration that this organism can do the same thing in a fermentative environment. Often we look at it as a thing that happens in the field, in a harsh environment where there's not enough nitrogen, not enough nutrients. Fermentatively, we have a lot of carbon, and we're showing that this organism is capable of fixing its own nitrogen. So nitrogen could be a rather source of ammonia via nitrogen fixation than using Haber-Bosch obtained ammonia in fermentation process. We had to go further to look at the genome of the organism, and clearly after sequencing it, we saw that it has a full repertoire of the nitrogen fixing genes. So that's exciting. It has all of it there, and it seems to be working. We're still studying that to make sure that they actually express during this ammonia deficient growth. But ultimately, apart from that aspect, one thing we have to bear in mind is the cost implication of this. Based on the cost of ammonium sulfate, which is often used in this fermentation process, if you think about the cost of ammonium sulfate today and you're running a 1 million gallon plant and you want to make a protein via precision fermentation, using about 7 to 8 grams per liter, you would consume about $65 million per year making your protein. So there's a real potential here in the sense that we can use nitrogen fixation not only for environmental sustainability in terms of what the footprints will leave on the environment, but mostly from the economic standpoint as well, that we can eliminate the cost of most nitrogen sources via nitrogen fixation. And I think it's not just with this organism, there's a number of other organisms we've identified that can fix nitrogen, which we can use in a fermentative environment. So in summary, we think that cheap lignocellulosic biomass materials and nitrogen fixation have real promise for precision fermentation. Of course, we still have a few things we need to tweak here and there to make sure that this is going to become commercially viable down the road. We need to establish the capacity of this organism, not just to use some or most, but if possible, all the inhibitors we obtain so that we can now go further to ferment whatever protein or product we're interested in without limitation, as we saw in some of the results we presented. But also, I think there's a virgin terrain for nitrogen fixation in precision fermentation. Rather than relying on really unsustainable sources of ammonia or nitrogen fixation might be the answer going forward. On this note, I'd like to thank all the people that have worked on this. They do the hard, dirty job, and I get to take the glory and talk about the work they do. My research specialist, Dr. Santos, I thank him so much, and all my students in the lab, and of course, USDA, Wisconsin Alumni Research Foundation, and the University of Wisconsin-Madison for funding this study. So thanks for listening. I'm looking forward to your questions now.
Thank you so much, Victor, for that fantastic seminar. I learned so much. I was just incessantly scribbling notes the whole time. And really, really appreciate you taking us through these many dimensions of your research. For those of you in the audience who haven't yet gotten the chance to do so, feel free to drop any questions you have for Dr. Ujor in the Q&A box of your Zoom bar, and we'll prioritize them in this next portion. So the first question that we have from our audience is about the use of lignocellulosic biomass for fermentation as studied for second-generation biofuels. So they kind of talk about how the commercial implementation of this technology was initially quite limited in the second-generation biofuel context due to competition from petroleum, even though it was extensively studied. So can you comment on how you imagine this would be different for precision fermentation, and how, precision fermentation for alternative proteins might be able to avoid the same path as second generation biofuels in that regard?
Yeah, that's a very good question, actually. The reason why lignocellulosics didn't take over the specter with biofuels lies in what I just talked about, the inhibitors. We can get as much sugar as possible, but if you cannot efficiently make as much of your product as you're interested in, then you're not going to be economically viable. So that brings us back to engineering bugs, identifying bugs, screening for bugs that can efficiently remove these inhibitors. Having said that, I do know that a few plants have actually come on stream. Poet biorefineries now making ethanol using a sizable chunk of lignocellulosic to do that. I know Green Biologics as well is doing that. So there's a few success stories in that terrain, but the real challenge has been the fact that we can get as much sugars as possible, but we cannot actually ferment them because of the inhibitors. So if we can find a solution to the inhibitors, we should be able to use precision fermentation to make alternative proteins. That remains the real roadblock to fermenting lignocellulose-derived sugars.
Thank you for that response. Next question. So reconstituting nitrogenase gene clusters is notoriously complex. This attendee is asking whether the most promising solution to try and use nitrogen-fixing bacteria as chassis for fermentation, domesticating them for this purpose, setting up a synthetic microbial community, or is it really introducing nitrogen fixation in yeast?
Being a scientist, I feel like I can't say that we'll never reconstitute the nitrogenous or the entire nitrogen fixing machinery in another organism. I foresee a future someday when that happens, but I think in the immediate, the challenge or the focus should be on using naturally occurring azeotrophs, the ones that can actually fix nitrogen. And there's a lot of them that can fix nitrogen that we didn't even know. Now we're sequencing a lot of genomes, we're seeing that, oh, they can do it. We feed them nitrogen all the time, they'll grow anyway. But once you remove it, you realize they have the capacity to fix nitrogen and they're fermentative in nature. So there's a lot of bugs that are out there that are fermentative in nature, which can actually fix nitrogen. I think the focus should be on those ones. How can we now fine-tune them to make proteins of interest or ingredients for making alternative proteins of interest? That should be the focus right now. I foresee a future someday where someone actually takes that and gets it to work in yeast, but it's still going to be a long time to come. We've tried it. It's very, very difficult to get the nitrogen fixing machinery to work in a heterologous environment. So I don't know if that will work soon, but hopefully someday. For now, the focus should be on using naturally occurring nitrogen fixing bacteria as candidates for making alternative proteins.
Awesome. Thank you so much for that comprehensive answer. We have another question here about whether the inhibitors that are coming from the cellulose or hemicellulose portions, is that where the inhibitors are coming from, or is it coming from the lignin portion of the biomass?
Well, it comes from all of them. Fermentated and HMF comes from cellulose and hemicellulose, the sugars. Then likes of vanillin, seringaldehyde, 4-hydroxybenzaldehyde, and ferolic acid, they come from the lignin fraction. Usually much lower in concentration. The ones that come from the lignin fraction are very small in concentration. The big ones usually are your furfural and HMF at the most. They come from the sugars. You have a lot of sugar there. Once you break it down, they dehydrate and degrade to form those inhibitors. So most of it comes from the sugar fraction than from the cellulose fraction.
Got it. Another question here is about moist heat pretreatment that you mentioned as a potentially suitable solution for breaking down lignin and accessing the hemicellulose and cellulose. Is there a cost-effective solution that you could recommend for kind of achieving the same objective? And do you think there would be any undesired effect on the proteins through those alternate processes?
You mean the moist heat for breaking down the lignocellulose? I can't think of any cost-effective way to do it right now. Using high temperature, I say high temperature, about 115 degrees Celsius, which is about the same temperature we use for sterilization. So using that in the presence of low or high pH is the most cost effective. You can use renewable, so it's very expensive. There are some methods you can use, but are significantly more expensive or extremely slower, which again becomes more expensive relative to using acid or alkaline pretreatment at high temperature. So those for me are the cheapest ones available today. If you can get rid of the inhibitors, I don't see how that can affect the protein synthesis, because then you have sugars. There's a relationship between the sugars and the microorganism making the protein in this case. If they can grow well in that environment, they'll be able to make the protein of interest for you.
Okay, so this other question is about nitrogen fixation via nitrogenase being a very slow process and resulting in hydrogen as a byproduct inside of the cell. So they're asking, as the first part, why not use e-ammonia, which is a well-developed process and a sustainable one, to take the place of that process?
If someone says ammonia, ammonia comes from one major process in
Yeah.
society. There's only Haber-Bosch process is the only way we can make ammonia in society today. Chemists have tried so much to come up with something cheaper, something milder, something better, but it hasn't happened yet, so to speak.
Mm-hmm.
I think it's just thinking about the overarching sustainability down the road. If you're thinking about something that doesn't harm the environment, what you've shipped about 560 million tons of carbon
Yeah.
to make the ammonia. So when you have the ammonia, you don't think about how it's made. It's easy to chop it in there, which is what we do to ferment. But in reality, down the road, we're still going through that fossil fuel,
Mm-hmm.
going through the high temperature to be able to obtain that. We do know that the nitrogen fixing process is slow, but from the results we presented there, using a small amount of ammonia didn't seem to harm the organism, just a small amount, allowing the organism to make up whatever it needs to grow using nitrogen fixation. And in the process, in the one I presented here, we grew the organism in a nitrogen-rich environment. Nitrogen is about $10 per tank. It's very, very cheap.
Mm-hmm.
An anaerobic chamber. In the chamber, you just flush in nitrogen, allow the cells to pick up the nitrogen themselves. So they do their thing fixing the nitrogen, which is lot more sustainable than having to work with ammonia. So most of us have not been to a Haber-Bosch process plant, so we can't envisage how much goes into the process per se, but it's not particularly sustainable, so to speak.
Right. Yeah, I think that question was specifically, and this is not something I have subject matter expertise in, specifically referencing e-ammonia, which is allegedly a feedstock. It's kind of low emission electricity, water and nitrogen, produced using renewable energy. I don't know very much
Yeah.
about e-ammonia. I know nothing pretty much, but
The source of the energy is all
yeah,
sustainable. That dampens the cost, so to
yeah,
speak.
yeah.
But you still have to obtain hydrogen to do it. And hydrogen, at this point, we don't have a sustainable source of hydrogen, at least on a cost-effective volume. There are ways to do it, so to speak, but you can split water to do that if you use a sustainable form of electricity. But then you think about
Mm-hmm.
it, we still have issues with water and climate change and all that. Again, the way I
Totally.
look at it, you could combine that with what we're trying to do. There's not going to be one single magic bullet, so to speak. We could have e-ammonia to some degree, but if you reduce the amount you're adding by allowing the cells to fix some of it, then you reduce the overall input. Hence, the sustainability increases as a result.
Mm-hmm. Understood. Thank you. Another question here is about whether assimilating nitrogen from waste streams might be a feasible alternative. And they cite specifically, for example, wastewater from agriculture. Is that something you've explored?
Actually, we're doing some work on that. We are looking at ammonia from wastewater. Yes, that's a possibility for sure. There's a lot of agro wastewater that's so rich in ammonia. And I have a paper that's just been accepted right now where we're looking at sourcing for ammonia from agricultural wastewater. And we can, again, the same graph I plotted, we can show that the cells can use that very efficiently. So yes, that's another alternative. Sourcing for ammonia from waste is definitely a possibility.
Awesome. Super exciting. We look forward to circulating that paper once it's published. This next question is about whether you can speak to what some conventional approaches have been to handling the inhibitor debacle.
You can use chemical processes to remove the inhibitors. That's one way to do it. And it's efficient, I have to say, in terms of how much you remove. But also it takes sugars as well. The processes also take sugars at the end of the day. So in terms of yield, if you spend time obtaining about, say, 100 grams per liter glucose and arabinose and xylose put together, and then you lose about 25% of that, then the process is no longer economically feasible because you're losing a fair chunk of the sugar you spent so much money to obtain. So there are chemical processes for removing the inhibitors, and the work But the reason they are not used on a large scale today is that they're not really feasible economically. They also lead to a significant loss of sugars.
Got it. Thank you for that breakdown. Um, there's a callback to your paper that'll be coming out soon, which we're very excited about. Can you share what species was engineered to assimilate wastewater?
Actually, we used a fungus to do that, Aspergillus delimar. We grew it in the presence of sugar and did not supply any ammonia or phosphate, so to speak. So we supplied the ammonia and phosphate and minerals from agro wastewater. And we asked the question, can this organism obtain the rest of the nutrients from the agro wastewater and then grow on the sugars? And it did work very well. In that case, we were measuring fumarate as a product to assess the growth of the organism, and we weighed the cell biomass at the end of the day. And we didn't see a significant difference between the wastewater one and the control. So it does
Wow.
grow on agro wastewater.
Wow, that's huge.
Yeah.
There's a question here that is just zooming out a little bit as you look at the potential for this technology to really help us build a more sustainable food system. You've talked about some bottlenecks, sustainability related ones, techno-economic related ones. This attendee is asking whether, as you weigh, the challenges, technical and economic, do you have a sense of, if you could just kind of synthesize what you think are the major bottlenecks when it comes to the technical feasibility and then the economic feasibility of making this happen at scale, maybe outlining, and I know this is becoming a massive question to answer, but outlining, what you think the roadmap might look like, to make this actually sustainable and scalable.
I think it's going to involve really, I would say, friendly economics and friendly environmental footprint. That we have to look at it from both ends, finding ways where we can cheaply get carbon and nitrogen. And if we do that, I think you can solve one problem with several problems with one solution. Because if you can cheaply obtain carbon and use them efficiently, if you can cheaply obtain nitrogen and use them efficiently, cheap and sustainable actually go hand in hand if you can do that. So let's say you can find a way to use lignocellulosics and it doesn't really limit the production of protein or any other product you want to use to formulate the protein. And also you you can obtain nitrogen either from waste and it's cheap and sustainable, always available. You're solving two problems in one. So I think if you make protein cheaply enough, people will buy it. That's what I believe in. If it's good protein, it tastes good, people will buy it. So what I believe we have to do going forward is looking at ways, that intersection between economics and technicalities. A technical solution that reduces the cost of making alternative proteins is what I believe in we have to be looking at. And that will involve looking at different things. And I don't think, like I said, one magic bullet that will solve that problem. There's a lot of research generated in society, which I think next week's presentation is going to be exciting. Looking at side streams, some side streams also contain nitrogen in addition to carbon. Which ones can we efficiently store and use? And I remember giving a talk about waste some years, about 2 years ago, and someone said, I believe in this way. The problem is, how can we store it and use it? So you can store sugar very well. How can we store waste and use it over time? Because it degrades in storage. That is where we really need to focus on. How can we build out the technology for storage so that some of the waste available can be used in making alternative proteins? Because that will allow us to reduce cost ultimately. And waste is cheap, waste is sustainable, so to speak. That's the point I want to look at. Really being smart in terms of the feedstocks we put into the process will help us achieve that goal of sustainability environmentally and economically as well.
Absolutely. Very articulately put. Thank you so much, Victor, for this presentation. I found it genuinely fascinating, and I think it will be of great value to our scientific undertaking here. Before we wrap up today, I'm going to give you an opportunity to share any last words or sentiments with our audience today. Is there anything you'd like to leave off on? We have a really collaborative community, so if there are specific research questions or bottlenecks you're experiencing, this is a great opportunity to do that. If you just want to leave this sector with some thoughts on where we really have to go in this road ahead, feel free to speak to that as well.
Thank you very much, and thanks to everyone for being here. I like to say that I tell my students all the time that just take a look at the summer every year. You realize that something is really happening. And if something is really happening, what's going to happen 10, 20 years down the line? So we have a responsibility as a society, as scientists, to find solutions to some of our most teething problems. How can we make proteins economically and sustainably? How can we produce things economically and sustainably? I think those are questions that not just scientists can answer, but all of us, no matter what you do as a person, there are many things we can do in our private life to help stem the tide of climate change. And seeing young people and old people and middle-aged people working on it excites me, and I hope everyone continues in that trend so we can find a solution to making proteins. As you say, making alternative proteins, not alternative. I like that quote a great deal. But most of all, my work in my lab, we look at a great deal of microorganisms, from nitrogen fixation to carbon sources. We like to engineer bugs a lot. If you're interested in things like that, I like to collaborate along those lines. I love waste a lot. I really am a waste guy, so to speak. Anything that is waste, can we valorize it? Can we add value to it? And if you're thinking of something you're working with that looks absolutely yucky, that nobody wants to work with it, those are the kind of things I like to look at, not necessarily always for food, but also to make all kinds of products. And if you have solutions, if you have ideas and thoughts along the lines of collaboration to convert something that is worthless today into something that is going to be worthy tomorrow, I'm interested in that area. And I think microorganisms and some chemical processes will really solve some of these problems going forward. So I'm excited about those solutions as we look into the future and continue to tackle this problem.
Thank you so much. That is such an inspiring note to leave off of. We've got a whole lot of work to do together as an ecosystem, but I think the path forward is bright. And as you say, ensuring that we have a habitable planet for generations to come and one that is able to really nourish all of our communities is an all hands on deck undertaking. So appreciate you for devoting your research to this mission and to all of the members of our scientific community for your contributions as well. Thank you so much, Dr. Ujor, for joining us today. Thank you for your work. Thank you to everyone around the globe who's tuned in today. And we look forward to seeing you again next month for a seminar with Dr. Alejandro Marangoni from the University of Guelph. All right, everyone. Thank you so much. Enjoy the rest of your day. Thank you so much, Victor. Much appreciated.
Real pleasure.
Bye.
Bye.